Welcome back everybody to our mini review sessions here with me, Arif Fazell. Now the next set of topics we'll be discussing is topics 26 to 29. For those of you who aren't familiar or keeping up with the topics, so those who are not in the class but are still listening and viewing my review sessions, which again I appreciate. So topics 26 to 29 is going to be discussing the cytoskeletal proteins, so various filaments that make up the cytoskeleton. The cytoskeleton is exactly what it sounds like. It's the skeletal structure of the cell. Now the skeletal structure of the cell is composed of various filaments, such as intermediate filaments, actin filaments, and microtubules. We will be discussing all of these filaments over the next few episodes. The one that we're going to start out with, though, is the actin filament. So the actin filament is one of the most abundant filaments in the body and in cells. Now actin can serve various roles, but one of the main roles that actin serves is a structural role. That is, it helps to support and maintain the shape of the cell overall. Now one of the first couple of experiments that I want to cover, that is again from topic 26, is discussing the actin filament and how actin actually contains an intrinsic polarity. That is, it has a plus end and a minus end. But the first two experiments that I'm about to discuss went ahead and discovered this intrinsic polarity. So trying to figure out exactly when you're looking at an actin filament, how do you know which end is plus and which end is minus? Now this polarity has nothing to do with charges, but rather where growth and subtraction of the filament occurs. So what I mean is this, so actin, remember, comes in two forms. There is the G-actin, which is simply the individual monomers, and this G-actin can then form and establish an actual filament. Okay? So this would be your G-actin, which is, again, stands for globular because it looks globular at the molecular level, versus the F-actin. So what I've illustrated here is that the G-actin can come together and establish and become a filament, or the F-actin, and then the F-actin also has the ability to depolymerize or break down back into the G-actin form. So what we're going to be looking at, though, is we're going to be looking at a filament of actin. And what we're going to try to figure out then is what is the polarity, or which end is positive and which end is negative. And again, this polarity has not to do with charges, but where do we see growth of the filament, so where we have new G-actins binding to in order to grow the filament, versus the minus end, which is where we will see depolymerization. And again, I'll also mention why we have depolymerization on one end and why do we have polymerization, or addition of G-actin monomers, on the positive end. So to initiate this experiment, we need an established F-actin, that is a filament of actin. Represented here by our straight line. Now the first half of this experiment, in order to determine the polarity, that is to try to figure out which end is plus, which end is going to be negative, we needed to use a protein that would help us actually visualize this growth and falling apart, or this depolymerization and polymerization. So in order to do that, what we chose was a common protein that we know interacts directly to actin. The protein of choice was myosin. Again, in the next couple of episodes, we're going to see what exactly myosin is. So, spoiler alert, myosin is a motor protein. That is, it actually has the ability to bind directly to the actin filament, and this occurs in the skeletal muscle, as well as smooth muscle and non-muscle cells, as we'll see. So what myosin does is it has a head group. It has a head group and then a neck region, this long neck region here. And it turns out that myosin, which is a motor protein, which means that it binds to actin, and then when activated by ATP, which we'll see later on, has the ability to detach from the actin filament and actually move or take a step forward in a certain direction. So this myosin starts out in one position on the actin filament, detaches when activated by ATP, ATP gets hydrolyzed, and now this myosin will actually then move to a new position on the actin filament, so somewhere further up. So again, we're going to utilize this protein as a way to help indicate or help us figure out which end of the filament is positive or plus and which end is minus. I'm going to start out with an actin filament as such. Now, we don't need the entire protein of myosin. As you remember from the drawing, myosin contained a head and a long neck region. The only part that we need is the head region. So in order to cut off the head of myosin, you can simply treat this with a protease. The protease that you can use is called papain. So papain is a protease that's actually found in meat tenderizers and will cut off the heads of myosin. In meat tenderizer, this causes the meat to kind of be loose and a little bit more tender. Again, we're going to use just the myosin heads. Now it turns out when myosin binds to actin, it binds at a slight angle. So what we're going to see then is I will actually see these head groups, and I'm going to represent these by red bars now, or red lines, and they bind at a slight angle. Just like this. So in black is our actin filament, and in red is the myosin heads, and they bind at this angle. So I have a myosin here, a myosin here, a myosin here, a myosin here. Okay? So that's 6 myosins, 3 on this side of the actin filament, 3 on the top side, and they all bind at this particular angle. As you can see, this resembles a spearhead or an arrowhead. So this is a point, so we will obviously call this end the pointed region, and these back ends here of the flailed ends called the barbed end. So barbed end, pointed end, barbed end, pointed end, barbed end, pointed end. Okay, so this is the first step of the experiment. So by decorating this actin filament with myosin heads, also known as S1 fragments, we will now figure out and establish which end of the filament is positive or plus and which side of the filament is minus.